Hi, my name is Eloise Fadial, and I work in Dr. John Oakey's Biomedical Engineering Lab. Today I'm going to talk about the fabrication of cell response microfluidic chambers with optimized fluid exchange. So a little bit of background. Microenvironments have become increasingly important for the study of cells. This has been true in the fields of cell biology and medicine, but also in more technological fields like they're used for inkjet printheads, DNA chips, lab-on-chip technology, and more. But microfluidics are a type of device that provide these microenvironments, and they have been becoming increasingly more popular. So, what is a microfluidic device? Well, there are these very versatile devices. You can create any 3D image within them. The ones that we use in our lab are made from polydimethylsiloxane, PDMS, which is a polymeric organosilicon. It is inert, it is non-toxic, and it is non-flammable. Another important aspect of these devices is that they operate on a micron scale, so you can really look at individual cells one at a time or other particulates one at a time as they come through the device. So this is one of the ways that we make microfluidic devices in our lab. So we begin with a silicon wafer. We pour a resin over that wafer, and then we have a mask that has a cutout of the image that we want to be sort of 3D printed onto this device. We then shine a UV light through that image, and anywhere that the mask has a hole in it, that resin will harden under the UV light. This creates a 3D image that is our new master device. Once we have this master device, we can use that to create as many imprints on the PDMS as we want. So we begin by pouring the PDMS onto that master 3D printed device. We then, after about an hour or so in the oven, or about 3 days not in the oven, the PDMS will cure and we can pull the PDMS off. We then take this PDMS layer and we put it under a plasma cleaner with a glass slide. This creates oxygen radicals on both the device and the glass slide or a glass cover slip. And when we put those together, they bond irreversibly. So we now have a chamber that can hold cells that will not change really in any way other than the tendencies of the PDMS. So it's very important, obviously, that we have a 3D design that we can start to work with. And this is kind of where my project comes in. So we already had a device that we were using to look at moss. And it looked very similar to this device here, figure A, except it did not have these two different layers. And the problem with this original device was that when we tried to switch out different fluids within the device to see how the cells would react, the fluids had a tendency to flush the cells out of the system. So I wanted to design a device that had traps, so it would prevent the cells from escaping the device, but also had a secondary layer where excess fluid could fill up that chamber and then slowly diffuse into the device, ideally preventing the cells from being flushed from the system. So these ones over here on the left are some of my models that I came up with, and the final model that I came up with is over here on the right. And this middle chamber here is 30 microns thick. The cells would be inserted into this larger chamber in the middle. They would then diffuse outwards, but would get caught by these little squares here, keeping them from leaving the device. The next inlet would be over here, and this would be for any fluid that you wanted to put in. This middle section could also be used as an inlet for fluid. And then, since this little chamber in here is 30 microns thick, this outer chamber is 250 microns thick. So any excess fluid is going to fill up one of these thicker chambers before slowly diffusing into the middle. I also was a little concerned about these two fluids clashing as they traveled around the outside of the device, so I added this separator here so the fluids from one side to the other don't meet until they are exiting through the outlet. So this should hopefully prevent any bubbles from occurring within the device. So my device, since it has these two layers, is really difficult to make using standard lithographical methods. To line up the masks exactly one on top of the other so that you can set these two different layers is nearly impossible. So I opted to use a 3D printer to try to print this device. This provided some of its own challenges since the most high-definition 3D printers we have at the University of Wyoming can print to a 30 micron scale. And so that's obviously the thickness of my smallest layer. And so it did take many trial runs for me to get some devices that were up to standards. So the 3D printer works very similarly to the lithographical methods that I described previously. This bottom section here, this little plastic thing, is filled with a resin. And as you can see in this picture, a UV light shines through the resin. A glass slide is put on this metal piece right here, and it's dipped into the resin. The UV light shines on it, and it does one layer at a time, creating each layer of the 3D device. Eventually, you have a completed printed device that hopefully works. So here I wanted to show kind of an example of how I test the device once it has come out of the 3D printer and I've made the PDMS model. So before I bind it to that glass slide, I take it and I look at it under a 3D confocal microscope. I use this microscope. It is called the LEXT, and it shines a laser and it measures the depth of your device. So here on the left is a very good example. We can see here the laser is shining. It stays at a relatively same thickness, and then it hits this section here, which should be 30 microns thick, and it drops down, which is exactly what we wanted it to do. And then this is where it would be bonded to the glass slide again, so it rises up, and then it drops down again once it hits this layer, showing that there's that space there, that cavity that the cells would be flowing through. Here in this image, this is my outlet here. My outlet, I've had a lot of difficulty getting to come out successfully because it is very narrow, and the PDMS sometimes tears when you try to remove the device from that 3D printed model. So this is just an example of one of the times that it has torn. As you can see, that middle section here, it's fine until you get to about right here, and this matches up with the image here. You can see it tore. There's all this kind of funky business going on down there. Once my device looks successful underneath the LEXT, I then use a rhodamine dye, which fluoresces under green light, to look and see how the dye diffuses into the device. So this is an example here. This is post my experiment. This is the inlet here. This is the outlet. As you can see, it went around. You can see there's some of this red dye in the middle, and that is where that dye has diffused in, and it's hitting those blocks, getting trapped in some of those spaces. But it does still come out in the end, as you can see here. This is another image there. This is at time equal to 30 seconds. I've just filled the outer edge with the dye, and this is at time equal to 10 minutes. There's still dye in that outer edge, which is good because I did fill it up with a lot of dye, but it has slowly diffused in. You can see it's a little lighter here, and the most important thing is this is the inlet punch right here in the center, where you'd be inserting the cells, and the dye, as you can see, has reached that hole punch. So this device has been a lot of work, but I have, I think, successfully designed and fabricated a novel device which contains this extra layer to hold excess fluid, as well as these traps to keep the cells within the device. I am currently working on calculating the exact diffusion rates within the device. And I would also like to see if I could change the outlet, maybe make this section here a little wider so that it would be a little more successful when I take these devices out. Right now I can still get a decent amount of them to come out successfully, but it's only about 40%, and optimizing device production would be ideal. All right, thank you for watching, and let me know if you have any questions.